Now it's time for a new, longer presentation with Florence APPARAILLY. Thank you very much to be with us today here in this studio. So it's better when it's in live. And as I said a few minutes ago, it's the occasion for all of you to ask your questions directly on your screen, on the bottom line Q&R of your screen. And it will be, of course, the occasion for you to know more about what you're going to tell us. And I will relay these questions after your presentation. Florence, you are Research Director at Inserm, Montpellier University Hospital Center of Montpellier, and you're a member of the Regenerative Medicine and Biotherapy Medicine Institute. And you're going to talk to us about control of monocyte diversity and plasticity by microRNAs for clinical application in inflammatory joint disorders.
Thank you very much, and thank you to the organizer for giving me the opportunity to be here today for this first hybrid event. And yes, it's been now 15 years that together with my team we are working to try to understand how microRNA control monocyte diversity and fate in the context of rheumatic disease and more specifically inflammatory joint disorders to design targeted therapies. Just as a short introduction, rapidly to explain you and remind you what are microRNA. They are small non-coding RNA that usually negatively control protein encoding genes by targeting messenger RNA into the cytoplasm of our cells. And this recognition is done on the basis of complementarity of sequence with the target mRNA. It is possible to introduce exogenous small interfering RNA, which are called siRNA, which are on the right part of this scheme. Those exogenous siRNA will interfere and use the mere machinery, cellular machinery of the cell to also silence target mRNA. The difference in between both is that microRNA recognition of their target is only a partial homology of sequence, while siRNA is a complete homology of sequence. But both end up by inhibiting protein expression of the target gene. So you understood that today I'm going to talk about rheumatism, and you heard before that a lot of money is put on rheumatic disease, but only 30% of patients answer to those drugs. So it's still important to do some work on that. Very rapidly, there are a lot of rheumatic disease, 200 different entities at least, and of course we are not addressing all in my team. I will tell you later. Although you don't die of rheumatic diseases, they are a cause of high morbidity and disability. And it's a huge burden, not only for the patient, but also for the society and our healthcare systems. It costs a lot of money. And it is in France approximately 16.8 million of people affected by rheumatism in France. So, and it's projected to be 78 million of US adults by 2040. The etiology of those rheumatic diseases is obviously very diverse. And the treatments are symptomatic because they address the pain of the patient and the inflammation, which is linked to this pain, trying more or less successfully to protect the joint function, but not very much success. And at the end, it ends up by destruction of those joints. So the bottom line is that there is no cure, so we still need to work on those diseases. And my team is working since more than 20 years on the physiopathology of a specific class of rheumatic disease, which I will call arthritis, which are chronic joint inflammation, to develop, as I told you, targeted therapies. To do so, we rely on translational research project, which goes back and forth to bench to bedside. And rely on good and well characterized cohort of patients, as well as cellular and animal models, to identify key regulators of joint homeostasis. we are studying 3 forms of arthritis, ranging from the top left from rare monogenic diseases that affect children and which are called autoinflammatory disorders, to more common adult form of arthritis, which is a rheumatoid arthritis, which is a multifactorial disorder. We also study juvenile idiopathic arthritis. The common denominator to those disorders in terms of immunological cells is the innate immunity which is involved, and more specifically monocytes. And why we think in our team that it's important to study monocytes in those joint pathologies, it's because they are really key. As you know, they can differentiate into dendritic cells and macrophage, which obviously drive inflammation, but they can also generate osteoclast. They can be differentiated into osteoclast. And osteoclast is the only cell of our body that is able to degrade bone, to perform bone erosion. So you understand that monocytes contribute to both joint inflammation and joint destruction, joint erosion. And so we try, with the prism of microRNA studies, to understand which genes are important to decide of the fate of the monocyte, whether they will differentiate into macrophages or osteoclasts, to play on those two features of the rheumatic arthritis. So this scheme is just to remind you that monocytes, they circulate in our body through the bloodstream. They derive from progenitors in the bone marrow. But what makes it complicated is that they exist in different flavors. And here I put you the 2 main flavors with the mouse markers, but it's the same in human. We have the non-classical LY60 high monocytes and the classical LY60 high monocytes and the non-classical LY60 low monocytes. And what we have shown in the past in the team is that arthritis conditions in the mice is associated with a huge blood monocytosis of classical LY60 high monocytes that will massively infiltrate arthritic joint, where they will be able to differentiate into inflammatory macrophages and or osteoclast. And so I will show you some data about microRNA controlling those fates. And it's very important to understand what drives this, their behavior and differentiation and heterogeneity outside and inside the joints. So you understood that our working hypothesis since many years is that if we succeed to target the macrophage osteoclast axis, we will be able to treat inflammation and bone erosion in those rheumatic diseases. But you have 2 problems to solve, or to address at least. The first one is that, as I showed you, monocytes are heterogeneous. So you need to have a system to deliver whatever drug you need in the right cell subset. And the second one is you have to identify the gene that plays a key role in the commitment of the monocyte to either macrophage or osteoclast. So I'm going to start with the delivery system. If you remember, I told you the LY60 high monocytes are the ones which are massively amplified in the blood of arthritic animals and infiltrate the joints. So we collaborated a while ago with a team of Daniel Sherman in Paris to develop a delivery system, which is a cationic liposome that's synthesized, that help us to formulate RNA interference molecule and to deliver whatever siRNA or microRNA we needed to LY60 high monocytes. And that's what you see here in green. Upon IV injection into mice, the LY60 high monocytes have uptaken the fluorescent siRNA, while the LY60 low did not. And more importantly, we can find back this fluorescent molecule into the joints of inflamed arthritic mice, at least 20% of them. What about silencing? It's good to uptake, but you need also to silence your target. Here I give you one example. We have been silencing NAMP, the other name is vispatin, which is a gene controlling cellular energy metabolism. And you can see on the top, when if you follow the blue lines and the blue bars, that mice that were injected with lipoplex formulated with siRNA against NAMP, the expression of NAMP protein was reduced in the LY60 high monocytes of the injected mice, while it's compared to in mice injected with a control RNA, interfering RNA molecules, while the expression of these genes remained unchanged in the LY60 low monocytes. And the consequence of this, it's a lower part, is that those LY60 high monocytes express much less TNF, the pro-inflammatory molecule that play a huge role in arthritis, both in the blood and on the bottom right in the macrophages derived from those cells into the joints. And more importantly, we were able to demonstrate in a curative setting that the repetitive injection of such a cationic liposome formulated with the anti-NAMP siRNA was able to efficiently stabilize the clinical severity of the disease. We are working here with collagen-induced arthritic mice. You can think about inhibiting any type of genes that are known to play a role in inflammation. For example, here, if you look at the orange curve, we are targeting TNF-alpha with siRNA lipoplex injected IV to arthritic mice. You can also think about combining siRNA targeting different types of genes, and here you see a mix of anti-IL-1, IL-6, and IL-18 siRNA. That showed a very nice inhibition of the progression of disease severity in mice. So, okay, we have a good delivery system. What about the genes? A while ago, we designed this strategy to try to identify microRNA that control the fate of monocyte, either to macrophages or osteoclasts. So we sort the cells, classical and non-classical monocytes, from the blood of human and mouse subjects. We perform a transcriptomic analysis based on the microRNA only, which is a miRNome. And we decided to study only the microRNAs that are conserved between both species and to perform in vitro and in vivo studies. So I have two stories to tell you very shortly. The first is miR125B, and it's in human. We've showed that the expression of this microRNA is highly expressed in the classical CD14 plus monocyte in the blood of human, as compared to non-classical CD16 plus monocytes. And when you look at rheumatoid arthritic patient, the CD14 plus monocytes, circulating monocytes, have a reduced expression of this microRNA. So we wonder what is the role of this miR in human monocytes. And to make a long story, a 6 years long story, very short, with this scheme, we were able to show that the miR125B in the CD14 plus classical monocyte was controlling their differentiation into and their polarization into inflammatory macrophages, synthesizing and producing TNF-alpha, as you can see on the right. And the miR125B did that by silencing two genes that are involved in mitochondrial metabolism, BIC and MTP18, which respectively controls the respiration of the mitochondria and the dynamic of the fusion and fission of the mitochondria. So miR125B obviously is a pro-inflammatory miR in those cells that can be targeted. The second one is a mouse story. It's miR146A. miR146A is the most highly differentially expressed miR between classical and non-classical monocytes, both in mouse and in human. Here are healthy conditions. It's much lower in classical monocyte than in non-classical monocyte in both healthy individuals. And our data showed that this low expression is even more reduced under arthritic conditions. So we knew at that time when we found out this that miR146A was a negative regulator of both inflammation and osteoclastogenesis. So we figure out that if this break is missing, we could use our delivery system to overexpress this smear in the LY60 high monocyte of the mice with collagen-induced arthritis. And on the bottom left, I show you again that we were able to upregulate the expression of this smear in the LY60 high monocyte upon IV injection of our formulation, cationic liposome formulation, when we formulated with mimic MIR146A, while the expression in the non-classical monocyte was not modified. And we didn't see any impact on the inflammation, but we did see a very nice reduction of bone phenotype in those animals, as you can see with those two readouts, which are on the left, the staining of the trap positive osteoclast within the inflamed joints that you are in the tissue and you can count. And on the right, the staining of the safranin O fast green, which gives you an indication of the quality of the joints. And on this model too, which are mice deficient for MIR146A, I wanted to show you histomorphometric parameters, and you have to look at the light gray bars that show that you restored in those mice lacking MIR146A, thanks to the liposome formulation, you restore the trabecular thickness and the bone surface of the mice, even if arthritis was induced to this mice. So, as a conclusion, I think I convinced you that we succeeded to identify targets, MIR targets, but also mRNA targets, involved in the pathogenesis of joint inflammation, to develop also RNA interference strategy to interfere either with inflammation or bone erosion, and to establish the proof of concept in preclinical models of arthritis that siRNA or microRNA can be of great potential for therapeutic use. My last slide is very short, but with the COVID pandemic, we heard a lot about RNA to be used for vaccination. And I want just to remind you that RNA-based therapeutic can be used as a strategy in other diseases and not necessarily as a vaccine, but typically I hope I convince you in rheumatic diseases, whether you need to repress or overexpress a protein of interest, you can use RNA interference. Thank you for your attention.
Thank you very much, Florence APPARAILLY, for this excellent presentation, which was really surprising by some, because microRNAs are not that famous, but you said indeed in conclusion that with COVID we heard a lot about it and of its capacity and capability to help in treating diseases. So I think we were convinced of the strategy for rheumatisms, and microRNAs are really promising for this kind of disease, but how your knowledge about the control of macrophage functions by these microRNAs can be helpful in other diseases than rheumatism, because you said it can be applied to other diseases.
Yes, indeed. As you can easily see, I talk about bone erosion, but also inflammation, and a lot of diseases are characterized by inflammation, typically infection. We were talking about COVID, so typically infection. So the target was that we have a pinpoint in our diseases or any type of disease driving inflammation. Can be of interest, and I want to say that we are currently working with a German pharma, Evotec, on those identifying MIRs in macrophages that can play a role in tuberculosis infection, for example. So the range of application is huge.
So with the COVID, we could see that the use of microRNA can be helpful, it works, but in the case of interfering RNA for monocyte and macrophages, because their lifespan is much shorter, it's very short. So is it really realistic to use that approach, that strategy for therapeutics, for treatments?
Alors, it's true, if you had asked me this like 10 years ago, I would go for more a long-term strategy. But now I'm convinced that it's a repetition of those the chronicity of the inflammation, so the repetition. So when you take aspirin because you have pain, you take one pill and you take the pain away. So why not injecting RNA interference just when you have inflammation to suppress this inflammation? So I really think it's not a problem. And I showed you systemic approach, but it could be local approach too. And I want to say also that now, thanks to single-cell RNA-seq and FET mapping in the mice, we have very exquisite tools. We know that those infiltrating monocytes, once into the joint, in fact, some of them remain for months. Okay. So, and they integrate the niche of the synovial tissue. So I think it's very important, even if you just target them on a short time, they will uptake whatever is a RNA interferent, and they bring it into the joint, and then you can change how the niche will interact with those inflammatory molecules on the more longer term.
So it means that we already have infiltrations to help in arthrosis specifically. So it means that your approach could be the same for local and short use, but it could be longer, it could be used longer, it could be helpful for a longer treatment, something that could be helpful for a longer time?
Yes, maybe we have had used our lipoplex formulation to inject intraarticularly, and we also found that we were targeting the LY60 high monocytes and the macrophage derived from them. So that's for sure, we can use it locally. After, of course, we will have to perform the experiments to know how long this will last, because we don't know yet, but
yeah, but to go back to arthrosis in itself, it's a very huge disease. It's very important in France. In many developed countries, it's a very important disease, and we don't have treatments, efficient and long-term treatments. So would your approach really be interesting for long-term treatment?
I think you have to think about it as a way to render the articular niche permissive to, for example, a tissue regeneration, because what is missing in rheumatic disease also is to restore, we can shut down the inflammation, but restore true functional cartilage and bone is much more difficult, and it's not done. So maybe by triggering a permissive anti-inflammatory environment for just a while to allow stem cells that you will be injecting to do their job and to regenerate cartilage and bone, it's something which is doable.
Something you can imagine. And are you close to clinics, or are you? What about next steps from now?
So talking about osteoarthritis, typically our institute, which is headed by Christian Jorgensen, is very much involved through a European consortium in part of regeneration of the cartilage using mesenchymal stem cells, and they have been in phase 2 trials on that topic. So we know now that you cannot regenerate a tissue in an optimal way if you don't control completely the inflammatory response into the tissue. So we have, of course, to talk together between people who want to make regeneration and people, immunologists, who want to control the inflammation.
Okay. Another question, which is just a detail, but I was very interested by the myrinome. Myrinome, because metabolome, of course, genome. We have so many nome and nomics, famous omics. What is myrinome really? Is it really something very well developed?
Yeah, it's an old name. It's a long time. It's you talk about transcriptome.
Yeah,
so transcriptome, it's also a transcriptome because microRNA are genes that are transcribed from our genome. So it's a transcriptome, but only for small RNAs. No, it's a true word. It's not a neologism from myself. And I even read recently a paper which was talking, I never heard before about it, of targetome.
Targetome.
Targetome,
Right.
looking at the targets of the mirror. Although you can create many words.
Right, and now we hope that one day we'll have the success home, which is only treatments and strategies with a great success. Thank you very much, Florence APPARAILLY, for
Thank
your
you very
participation,
much.
your presentation, and this very interesting question. Oh my God, I have a question. I'm sorry, didn't give a look on my tablet. So, do you expect any strong side effects by lowering the response levels of these immune cells? It means being prone to specific types of infection due to immune function loss in those tissues?
So of course you have to check that, but I don't think so, because those cells are short-lived, as you just mentioned before, when they are in the circulation, and they are rapidly attracted to the site of inflammation. So of course off-target effects have to be tested, but I wouldn't be afraid of that, because they really rapidly go to the joints and do their job. And once they are in the niche, they don't go out like that.
Okay, thank you. It was a question asked by Jonathan Despont from Health Data Hub. Thank you very much for those questions. Thank you, Florence, for your participation.